welcome Dr. Kester.
Good morning, everybody. I just want to take the moment to thank Martine and, more importantly, Temple and Genesis for making us all feel warm for 2 days. So today I'm going to be talking about another type of nanoparticle. I like to think of it as the green nanoparticle because of its bioresorbability, its lack of toxicology, and we're going to be talking about calcium phosphate as a nanoparticle. And we're going to be talking it as nanosolutions for bioimaging, for drug delivery, and now that I've got patent protection, I can now talk about it, photodynamic therapy. And in terms of full disclosure, I like to consider myself a faculty entrepreneur. I founded a company called Keystone Nano with Jim Adair, who is the director of the NSF's Particulate Material Center, and we are commercializing calcium phosphate nanoparticles. So I don't have to tell this audience what is nano, but I'm always impressed. This is before Obama's stimulus package, that the US government was spending $3 billion US annually on the National Nanotechnology Initiative. It's only going to get better, in my opinion. And obviously, we're exploiting new novel properties of nano-sized material. And I don't have to tell this audience that the future of medicine is small. In fact, it's really, really small. But what I do have to keep reminding myself of is that we've heard a lot of different nanoparticles over the last day and a quarter. We haven't even talked about nano-sized dendrimers, polymeric micelles, nanoliposomes, ferric oxide, nano-sized particles. There are specific limitations to what you can do with these materials. Everything that they may start, and the key one to me is agglomeration. You may start with a nano-sized particle, but as soon as it hits biological fluid, physiological buffer saline, or inside an animal model or human, it self-aggregates, it agglomerates, it becomes lunch for a macrophage. We also do not really talk about the size and shape differentiations, the ability to cause allergic reactions or immunogenic reactions, the ability of these materials to, once they enter the cell, to be pushed back out through ABC transporters or P-glycoproteins. The idea of loading efficiencies and how to maximize them. These are the type of things that we are considering as we exploit calcium phosphate. Because at the end, I want to come up with a bioresorbable, biocompatible particle which will allow us not only to image, and specifically optically image, but also allow for targeted delivery of insoluble agents and or molecular agents such as siRNA. I really don't have time to talk about two recent papers that came out in ACS Nano Letters talking about these materials as biosmart bombs. We have a pH-responsive trigger. You can create, and there's a nice representation of what our calcium phosphate nanoparticle is, but we exploit the properties of calcium phosphate. That pH 7.4, this is a relatively stable nanoparticle. But inherently, as you lower the pH below pH 6, calcium phosphate at this ratio dissolves. Out comes your active ingredient, and surprisingly, even though it's endocytosed or pinocytosed, when it dissolves as a function of going through the endosome, as a function of pH, your drug or your active ends up in the cytosol. That's just one picture of the calcium phosphate nanoparticle. You'll see a couple later that truly showing the non-aggregating in physiological solutions. And obviously, you can put different photoprobes in, and they come in different flavors and colors. The key differentiating point between this technology and other technologies, besides using calcium phosphate as a bioresorbable, biocompatible material, is that we truly cage the material. We notice, you're going to see in a couple of slides, that we actually take the active ingredient and we have that as part of the synthesis, so we actually wrap the calcium phosphate matrix around the active in real time. That leads to protection in biological fluids, and that's a major differentiating factor. So let's look at the nanostructure in a little more detail. The active, which is in green, is encapsulated, caged within the nanoparticle structure. We can then cap these nanoparticles. That allows them not to aggregate with each other, but we can put different surface functionalizations on the outside. They're about 1 nanometer in size, and they can be either negatively charged, positively charged, or highly pegylated, and we'll see that the highly pegylated forms are going to be very useful in biological animal models. We refer to them as calcium phosphate nanoparticles, but if I was talking in terms of Keystone Nano, we would call them nano jackets because they're dressed to kill. Marketing is everything. This is actually a scanning EM of the indocyanin green doped calcium phosphate nanoparticles. We're going to put a near-infrared agent within the calcium phosphate nanoparticle because we're going to have incredible bioimaging capabilities and/or the ability to do photodynamic therapy. Notice they do not aggregate. They're about 16 to 20 nanometers in size. This is my slide. This basically is the work that Jim Adair pioneered and that we helped bring to fruition. But basically, we make an inverse double micelle. We then mix the calcium phosphate materials together with the active. We add a little metasilicate, and then we use a dispersant to break it apart. And then we go through a capping procedure to make sure they stay the ability not to aggregate with each other. And the really neat part of the technology is down in the lower right, is the ability to launder these materials. We use HPLC as a way to purify and to enrich these materials as they go down the silica HPLC, and that way we can get high yields of these materials. We can make batch-to-batch reproducible calcium phosphate nanoparticles, and if you see on the right, as we put them in physiological saline solutions for up to 7 days, and in reality, these experiments have gone for months, they do not change in size. So they stay non-aggregating, they stay as 20 nanometer sized particles. We can create them in different charges, different surface functionalizations, either carboxylic acid, so we have zeta potentials of a -50, or we can put amines on the outside, and we have zeta potentials of +30. But the ones we're really interested in is the one in red, where we pegylate them. That leads to some incredible pharmacokinetic properties. That's the one with the 0 zeta potential. And that also, in all cases, we have the ability to put on small antibodies and small peptide fragments for more targeted delivery. Despite the ability to do this, and we don't really have time to discuss those studies, which are showing up in bioconjugation, we will show that just having a functionalized particle, just with the pegylation, leads to enhanced permeability, retention effects, and targeting of tumors without the sophisticated targeting. I want to spend the next 5 minutes just talking about some of these optical imaging capabilities of the nanoparticles. So we created near-infrared nanoparticles. We've doped the calcium phosphate nanoparticles with indocyanin green. This is a very sophisticated experiment. It's a laser diode we created in the lab, and a good optical camera that I bought at Best Buy in the United States. Put the right type of photo filters on the outside, and you can see we can actually see the near-infrared fluorescence. Let me first talk about the limitations of indocyanin green as a fluorescent agent in the near-infrared and the benefits when you encapsulate it. Whenever you use a fluorescent agent in biological fluids, you get dimerization, you get protein binding, and it's all going to lead to instability of the fluorescence material. And we talk about fluorescent stability, we're talking about in terms of seconds. You also get rapid elimination from the body. Indocyanin green in most models is cleared within 3-4 minutes. You're going to see images 4 or 5 days, and these animals are still lighting up specifically in the tumor. So we get 2 types of stability. One is a pharmacokinetic stability by putting it within the calcium phosphate nanoparticle, and the other is a stability of the fluorescent agent itself. The benefits of caging include the ability to keep these fluorescent materials as monomers, stop them from aggregating themselves, and when we actually encapsulate within the calcium phosphate nanomatrix, we can put them at specific lengths from each other so we don't get quenching and we don't get self-absorption. We can protect for photostability, and we can see tumor targeting by enhanced permeability and retention, as you'll see in the next couple of slides. When we look at the absorption and fluorescent spectrum of the doped ICG nanoparticles, notice the black curve compared to the red curve. The black is the fluorescence we see when we encapsulate. Notice it's greatly enhanced and shifted a little to the left. That's what we'll talk about when we measure quantal efficiency in a couple of seconds. One of the neat things is, if we just use indocyanin green, which is free indocyanin green, the curve on the left, As we change the environment is in water, DMEM, PBS, phosphate, sulfate, saline, notice the absorption and emission curves shift. It changes as a function of the environment. Look at the curve on the right. Now we've encapsulated within the calcium phosphate nanoparticle, and we put them in the different environments, and it's the same curve. Again, speaking to the fact that we have stability within these physiological environments. When we actually did fluorescent correlation spectroscopy with some of our colleagues, Peter Butler and Peter Eklund, who are true photophysicists, we looked at the fluorescent half-lives of these materials, and this has just been published in ACS Nano, for if you want to get some more information on how we actually did these experiments. But at 2 different power intensities, notice when we encapsulate the ICG within the calcium phosphate nanoparticle, we get about a 5-fold increase in the fluorescent half-life of these materials. Also, when we did fluorescence correlation spectroscopy and looked at quantal efficiencies, we see about a 2 to 2.5-fold increase in quantal efficiency by locking or caging the calcium, the indocyanine green within the calcium phosphate nanoparticle. That speaks to the power to do optical imaging. So these were not as sophisticated slides of galaxies, but to me, these are really neat. Let's just go down to the, there we go. Let's start here at 96 hours. These 2 dots right here, those are the tumors. And let me just work your way how we get there. So there are 4 animals. You're looking at the same animal as a function of time. We anesthetize the animals, and at either 15 minutes, 2 days, 4 days, we're taking pictures of the same animal. I apologize for that animal moving when we took the picture, so that's why he's not in focus. Notice we got 4 groups of animals. We've got ghost calcium phosphate nanoparticles, ghost meaning there's no indocyanine green within the particle. We've got free indocyanine green in the second animal, and we've got 2 different types of calcium phosphate nanoparticles, one with a negative charge and one highly pegylated. Notice the first thing you see is there's our liver, and we'll come back to that in a second. But notice over time, the material is being cleared and it's ending up in the fecal material. And in data not shown, and you can look at the ACS Nano paper, we actually have a really neat mode of excretion here. It's enterohepatic biliary recirculation. It's going to the liver, but it doesn't get stuck in the liver because of the size, ends up in the biliary circulation, ends up in the gut and out the fecal material. Really neat. But we still have enhanced half-life, and over time, we can see it accumulating within the tumors. Notice the free indocyanine green is cleared within 24 hours, but when you have increased ability, this increased half-life of the material itself, the calcium phosphate nanoparticle, it leads to accumulation within the tumor. And this is a good demonstration that enhanced permeability retention. These are not targeted material. They're only pegylated. It's been shown that pegylated materials have some affinity, because those pegylations look like sugar, for tumor cells. And it's a good example I've seen of enhanced permeability retention. If you're small enough, you're going to go into a tumor, but you're not going to come out, and you're taking advantage of the pharmacokinetic properties of the material. So we can image the tumors. And this is a really neat, sophisticated experiment. I mean, you can, again, use a very cheap digital camera here. And here we went to Costco and picked up a ham. And inside the ham, we put the indocyanine green materials at certain lengths and distances. And you can see here, this is free indocyanine green. Rapidly lose it. In this experiment, we went as far as 3.5 centimeters, and you still see the material. And now, with different ways we encapsulate, we're seeing 6 to 7 centimeters within biological organisms. We're trying to create optical imaging as a modality that will compete one day with PET scanning, MRI, et cetera. And we're taking advantage of the ability of these materials to have quantal efficiencies that are higher than normal because you cage them within the nanoparticle. So how can we exploit that? Well, that's just imaging. But you can take imaging and often make it kill. So this is photodynamic therapy. It's been used for tracheal cancers, esophageal cancer. Bottom line, it's used for those cancers because nothing else works, and photodynamic therapy itself really doesn't work because of the certain limitations to the fluoroprobe, et cetera. We may be able to get around that by encapsulating with calcium phosphate nanoparticles. So for photodynamic therapy, you need a photosensitizer, you need light, you need oxygen. You excite the photosensitizer. Here it's going to be a near-infrared probe. It's going to be our indocyanine green. We're going to excite it, and we're going to transfer that energy to molecular singular oxygen, which is going to be used to kill. But there are limitations to photodynamic therapy. including low quantum yield of the photosensitizers. We may be able to get around that through caging, through a paper that's coming out in Nano Letters the next couple of weeks. Psi 3 and Psi 5, we're seeing quantum efficiency increases of 7-fold upon caging. This limited by the short lives of the photosensitizers. We can increase that by having them caged within the calcium phosphate nanoparticles. And there's a possibility, there are problems with getting enough light within the organism. We can use near infrared, and that's a new advantage of photodynamic therapy. So our technology has certain advantages to allow this to happen. They're bioresorbable, biocompatible materials. The PEG allows for enhanced permeability retention. We can encapsulate the near-infrared fluor with these great new photostability and photoefficiencies, and we can penetrate tissue. We may have the pattern put together. So let's look at some real examples now. We're going to go to animal experimentation. These are not intratumoral injections. These are all injections into the tail vein. This is pure systemic therapy, the way it's going to be done in the clinic. Here we're looking at a breast cancer model. These are new models. There's an MBA 231, highly aggressive tumor cell. And the bottom line is, this is the indocyanin green calcium phosphate nanoparticle. All of these up here are either the controls, PBS, ghost nanoparticles, no indocyanin green, free indocyanin green, or the negatively charged material. What we do here is we go up the tumors over 7 days to a defined volume. That's the day 7. At day 8, we give the calcium phosphate nanoparticle. In data not shown, 24 hours is the key. That's where we start getting the enhanced permeability retention. We then take a good optical laser. We focus it in on the tumors. We give 3 short bursts of an intensity that's 1/4 what has been used in the clinic before, 3 15-second bursts, and that's it. That's the entire therapy. And then we start following the size of the tumor. And you can see, tumor's not growing. Neat. Let's get better. That's a solid tumor. That's cheating. Let's go after leukemia, non-solid tumor. Now you can start thinking non-solid tumors. You can start thinking Mets. And this data just came in last night. So that's why I asked Temple to put this in, but I think it's really neat. Same type of experiment, photodynamic therapy for an AML model, acute myeloid leukemia model. Here we're putting the tumor cells into the animal. We let them grow. They end up in the spleen engorges. We don't have time to show you that data. And basically, you can see increased white blood cells over a factor of time. Here, every 4 days, we're going to give the photodynamic therapy, the laser treatment. We target it against the spleen 1 day after we gave the calcium phosphate nanoparticles. Here are the tumors growing. The white blood cell counts are increasing. This is going to be a 4-day experiment, so this is halfway done. But you can see here is the control, and here are the different conditions. No white blood cell growth. So we can actually, and this again is tail vein injection of the calcium phosphate nanoparticles. Notice the other thing here is not only works for indocyanin green as a photodynamic therapy, but we also use one of my drugs here, a bioactive lipid that's selectively apoptotic for cancer cells. It's called ceramide, or for the French here in Quebec, ceramide. Ceramide is a bioactive lipid and basically is selectively autophagic for cancer cells. And you see the ceramide-doped calcium phosphate nanoparticles also prevent the growth of the tumor. We're starting to look at mechanism. And here we're looking at free reactive oxygen species. This is one experiment of several, but here we're using N-acetylcysteine, and basically we're looking at cytotoxicity. This is a very, very low dose of the laser. You can see we can become cytotoxic as we give the photodynamic therapy, but we prevent it with the N-acetylcysteine. These are some of the experiments we're asking. Is it reactive oxygen species that is the photodynamic therapy? Is it accumulation of ceramide? We're doing sophisticated lipidomics by mass spec. Are there other mechanisms that lead to the photodynamic therapy and the enhancement of photodynamic therapy by encapsulating near-infrared light, near-infrared photoprobes within the calcium phosphate nanoparticle? We can also do drug delivery. So besides photodynamic therapy using the infrared fluors, we can put drugs into these calcium phosphate nanoparticles. For a company perspective, you want to get to the clinic as quickly as possible. So even though we can do non-FDA approved drugs like ceramide, and we're working with the National Cancer Institute on some of their new drugs. In reality, to get to the clinic, you want to get there as quickly as possible, and you want to use already an approved drug. Here we're using doxorubicin, which is a cancer-fighting agent that's often used for women with breast cancer who are estrogen receptor negative and HER2 negative, very, very nasty cancers. We grew up and we created a doxorubicin-resistant cell line. By definition, doxorubicin's had these cells and it's pumped right back out. And we can see here, we can load the calcium phosphate particles with doxorubicin, and here at 10 to the negative fourth molar, you put free doxorubicin into the tail vein. Again, these are all tail vein injections, and nothing happens. Breast cancer cells are growing out of control. When we put 1/10 the dose, this is 10 to the negative fifth, 1/10 the dose of doxorubicin within the calcium phosphate nanoparticle, we see about a 40 to 45% decrease in the size of the tumor. So we're using a highly resistant, dox-resistant cell line, we're doing systemic delivery, and we're decreasing the dose by 1/10, and we're still seeing biological efficacy because of the encapsulation. And I'm going to just leave on this slide. We don't have time to show you how we can put avidin on the outside of these particles. It took a year and a half. I'm going to summarize a year and a half in about 30 seconds. We can put avidin on the outside of this material and put on any biotinylated antibody of choice and get really specific targeting. All the experiments you've seen up to this point were done without targeting. And the FDA likes that because, again, the only variable then, if you're going through phase 1 trials, is the nano itself. You're using an FDA-approved drug, doxorubicin, and there's no sophisticated targeting mechanisms. But we can make these particles better through the targeting. This is just a quick way of showing that we can do the targeting. The mascot of Penn State is a nittany lion. We have actually do some chemistries to allow us to put the green, in this case, nanoparticle, calcium phosphate nanoparticle, only on the gray areas of this very small stamp, and you can get very exquisite targeting. And we can do this not only in terms of a nittany lion stamp, but we can do it with cell tissues. So I just want to end right now on the calcium phosphate nanoparticles because of their biocompatibility, the bioresorbability, and the pharmacokinetic properties of these materials, and the ability to put a pH-responsive trigger within them, have the potential to deliver the promise of non-toxic bioimaging delivery systems. And the key part is it's bioimaging, it's drug delivery, it's photodynamic therapy. It's the ability to seek, treat, and image all at the same time. So with that, I'm open to questions.
Thank you. We have time for some questions. Who would like to question our speaker? Please, if you could come to the center here.
Simon Benita, Hebrew University of Jerusalem. Very nice talk, very impressive. I have, and we are going to publish exactly the same about the elimination of the nanoparticles with pegylated polyesters, because it's really something interesting. And I would like to have your publication to cite you because you're the first one before us. What do you think about this kind of elimination? Have you an idea?
Excuse me, I couldn't hear that.
Have you an idea how is this kind of elimination by the enteropathoscycle system is?
Honestly, we didn't expect to see the whole idea of targeting the liver and ending up in the...
We had the same surprise, by the way. We thought we are going to publish it in 2 to 3 months.
Right.
And we just repeat the experiment.
Exactly. It was a surprise to us, too, but it's a neat surprise.
Yes.
And I believe it has something to do with the size of our particles, that being able to maintain 20 nanometer-sized particles, that we're escaping the sinusoids and we're actually ending up not being eaten by the Edo cells, et cetera, and that we have found the way, these particles have found the way to...
Our particles are 100 nanometers, are different, and we have the same elimination pathway.
Again, it's neat, it's wonderful. So now we can start thinking how we can exploit that. So
yeah.
We end up in the liver.
Okay.
So now I'm starting to think, maybe I can use photodynamic therapy. Let's say we've got a pancreatic tumor and we're lucky to resect it. Maybe I can go in with a biopsy or go in with my laser light during the resection and go not only target the pancreas, which we know we can target too, but the liver and the lymph nodes. I mean, maybe the fact that we're accumulating there, maybe it's a way to go after Mets. And in data not shown, we've done all the liver enzymes you ever wanted to do in your life, and we're not getting any toxicology. So it's a neat thing. Sometimes things happen serendipitously. I mean, the photodynamic therapy, serendipitously, my lab was supposed to sacrifice some of these bioimaging animals after 4 days, and they forgot. We had multiple animal experiments, 30 days they came back and said, by the way, you're paying for 30 days more animal care, but the good news is there are no tumors. Sometimes you get wow. So.
Well, that was